Hello friends, welcome to my YouTube channel. Myself Janvi. Today I have come up with a part 2 of the topic on cell growth and cell division. If you have not seen part 1 of the video, please see the description for the link. Before I start today's video, if you like my channel and videos, please subscribe to it and share it with others. To summarize this topic or the course is made into 3 parts, so watch all the 3 videos to cover the topics. In the part 1 slide of cell division, we have learned a few things like what is cell cycle, what is cell growth, what are the stages which are involved in cell cycle, like it may be G1, S and G2 phase, and how the cell enters into the mitotic phase, like what are the cell has to go through to enter into the mitotic phase, like it may be a prophase, metaphase, anaphase and telophase. But in part 2, we are going to cover about a cytokinesis. What is a cytokinesis? That is nothing but a division of cytoplasm. Next, how the regulation of mitosis takes place. And what are the things which will play a very important role in mitosis? It may be a MPF factor or it may be a role of cytoskeleton in mitosis. And importance of mitosis. What are the main importance of mitosis? Once the nuclear division is completed, till whatever we have studied, like it may be a prophase, metaphase, anaphase, or the telophase, all this includes only a nuclear division. But we can say only the cell division is completed until and unless the cytoplasm is divided. So now we have to concentrate on the cytoplasmic division, which we will call it as a cytokinesis. So now the nuclear division has taken place and the cell is now ready for the cytoplasmic division. So in the future slides I am talking about the cytoplasmic division. Cytokinesis. Division of the cytoplasm is called cytokinesis. The spindle fibers play an important role in deciding when and how the division should take place. It starts in anaphase and continues till the telophase. Cleavage takes place through the plasma membrane with the help of the actin and myosin filaments. The contraction of myosin and actin filaments introduces a stress on the plasma membrane, thus it creates a furrow and divides the cell into two. In M phase, certain organelles like Golgi complex, mitochondria break up into smaller fragments, thus organelles are equally distributed into the daughter cells. Here we can see this is the plasma membrane. This blue color indicates the actin and myosin filaments. We can see that because of the contraction of the actin and myosin filaments, there is somewhat a bend which is seen in the plasma membrane. This bend or burrowing is brought about with the contraction of the myosin and actin filaments, and this burrows will get deeper and the cell will divide into two daughter cells. In the earlier slides, we have talked about the mitosis and the stages which are involved in the mitosis. But now we will speak about the regulation of mitosis by various factors. There are 3 important factors which will affect the mitosis. One is regulation by mitotic chromosome cycle. Second one is dissolution and formation of nuclear envelope during mitosis. Third one is role of cytoskeleton in mitosis. So we will explain each of this in a different slides. Regulation of mitosis and cell cycle. As I've already said, there are 3 factors which will decide whether the mitosis should take place or not. In that, first one is the regulation of the mitotic chromosome cycle. In this, we will explain what are the factors which will affect the mitosis. Like first one, S phase activator. Second one is M phase promoting factor. Third one is M phase delaying factor. The appearance and disappearance of these factors will play a very important role in deciding the regulation of the mitosis or whether the mitosis should take place or not. See, what is the role of MPF protein in controlling and regulating the mitotic cycle? See, as I've already said you, there are three factors which will help in controlling or regulation of the mitosis, and each are connected with one another. For example, see, MPF protein is the one which I spoke in the earlier slide. Here I am talking in detail about it. The MPF protein cannot enter the cell, or the cell cannot enter the mitosis until the MPF protein is produced. And the MPF protein cannot be produced until the M phase delaying factor has been disappeared. Till the DNA synthesis completes, the M factor and the S factor cannot disappear. And the DNA synthesis cannot complete until all the DNA replicates in the cell, and the DNA cannot begin to replicate until the block is removed by the mitosis of the G1 phase. And ultimately, a cell cannot progress from mitosis to G1 when the chromosomes are separated on the mitotic spindle. Now we will talk about MPF subunits and its function. MPF is a large sized protein and it has two subunits in them. One is inert subunit and another one is kinase subunit. Inert subunit will help in activation of the other molecules, whereas kinase subunit helps in self-activation of the protein. The chromosomal condensation will take place with the help of phosphorylation of the certain molecules including histone. MPF also triggers the breakdown and formation of the nuclear envelope. Most of the things which are regulated in the mitotic cycle is all brought about by the MPF protein, either by phosphorylating it or by dephosphorylating the molecule. If it phosphorylates, then the chromosome will get condensed. If it dephosphorylates, again the chromosome will convert back itself into a chromatin. These are few signals which are given by the MPF protein towards the cell. Dissolution and formation of the nuclear envelope during mitosis. The outer and inner membrane of the nuclear membrane disappears. The underlying nuclear lamina disappears. The nuclear pores are formed. The phosphorylation and dephosphorylation will play a very important role in developing and disappearing of the envelope. The pre-synthesized nuclear pores assemble and reassemble with the help of the phosphorylation. Hence, this all are taken with the help of the M promoting factor. Role of cytoskeleton in mitosis. They mainly appear in the M phase. The mitotic spindle formed helps in the migration of the daughter chromosomes towards the opposite poles. Contractile ring of microfilaments will help in the division of cytokinesis, that is nothing but the division of the cytoplasm with the help of the actin and myosin filaments, which we have already discussed. Meshwork of the intermediate filaments that surrounds the interphase nucleus will also include two daughter cells. Now we will study about the importance of mitosis with the help of the chart. Here you can see the importance of mitosis. It helps in maintaining the size of the cell and it maintains equal amounts of RNA and DNA. It helps in growth and development of various organs of the body. It helps in removing the old decayed cells and dead cells from the body and involved in asexual reproduction in few animals. Okay, just we'll fastly recap what we have studied in the part 2. Mainly we studied about the cytokinesis, that is nothing but the division of the cytoplasm. Then we learnt about the factors which regulate mitosis, either it may be a MMPF factor or it may be a dissolution of the nuclear envelope, or either you can talk about the role of cytoskeleton in mitosis. All these factors will decide whether the mitosis should take place or not. Then we learned about the importance of mitosis, why the mitosis will play a very important role in the cell division.